go ahead and get started. First of all, I just want to thank everyone for being here. Really wonderful to see you all, and I'm super excited about tonight's lecture. Just a second, not yet, almost. Anyway, yeah, my name is Leslie Thompson. I'm the co-director of the Stem Cell Center, along with Brian Cummings over here, who's the other co-director here of the center. And yeah, and so just a couple notes for the center. So just we're really proud of a lot of the work that's been going on in the center, and we really go from bench to bedside. We have the new Falling Leaves research building that you probably have seen when you came up from the parking lot, just right over there, that has a stem cell program in that sort of targeted toward neurological disorders. Then translational research, we're very fortunate here to have a GMP, good manufacturing process facility for cell and gene therapy, that we can actually make the products that go into humans. And then an Alpha Clinic stem cell, Alpha Clinic Clinical Trial Center here, run by Daniela Bota, that where we can take then these gene and cell therapies into the clinic. And we have a number of clinical trials here, with a few more hopefully coming up. And as I mentioned, the Alpha Clinic that is enrolling over 200 patients to date. A number of training programs as well. And just to highlight a couple recent things that have been going on here in the Stem Cell Center, Lisa Flanagan, who's one of our faculty here in the center, has been looking at communication between blood cells and brain cells, and what drives what's called neural stem cell development. And this has implications for stroke, for patients recovering from brain injury and stroke. And so there's on the site, you can read more about this work that just came out. And then also from one of our newer faculty, Herdeline Ordonia, who has been working on light-triggered cardiac therapy, which is super cool. It's really groundbreaking research, and you can read more about that on the stem cell center site as well. And so, you know, these are just a couple snapshots of some of the work going on in the center, but it's really an exciting place. And as we all know, the science really drives these kinds of translational findings and clinical trials and applications to patients. And there's a huge impact of science funding on progress to date. So just a few stats to remind people about the importance of science funding. But we have lots and lots of information on the stem cell site, on the Bill Stem Cell Research site, upcoming events, the website itself, people who are in the center. And tonight we're really fortunate to have two individuals from UCI who are going to give a tag team talk together. They're going to go back and forth, which is going to be really fun to hear. But both Xing and Christina Kraus have been involved in working on skin biology for many, many years. Actually, Xing and I go way, 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 way back in the Department of Biological Chemistry. She's a professor in the department. She's the vice chair of biological chemistry and the director of the molecular biomedical division within the School of Medicine, right? And has worked on skin disorders for many, many years. Her early work was in Elaine Fuchs' lab, who, for those of you who know that lab, really has been instrumental in mouse genetics. And Xing has laid a lot of the groundwork for skin diseases using mouse models, and then more recently using stem cell models. And she had her bachelor's in Shanghai in China, University of Hawaii. She had her master's, University of Chicago. Her PhD and postdoc work was also at the University of Chicago, and then came to UCI. And I've known her for a very long time, which has been really fun. And then Christina is an assistant professor here in the Department of Dermatology. She's also the director of the gynecological dermatology at UCI. She's the associate director for the dermatology residency program here. She did her undergrad at UCLA, her MD at Georgetown, her internship at Georgetown also, and you were the intern of the year there too, which is really cool. Congratulations. And I did a residency at UCI and was team president of a dermatology residency and her master's in public health recently at UC Berkeley. I'm trying to figure out how you did that while you were at this, okay. While you were doing all this. But really a pleasure to introduce both Xing and Allison, if you want to come on up. Thank you. And when you come up, you each have to say one fun fact that would not be in your CVs. And let us know something that you enjoy or whatever you want.
I'm gonna regret this, but I enjoy singing a lot.
Ooh, you are gonna regret that.
I didn't know this about Xing, and I'm so excited to know this. I have a black belt in Taekwondo.
Okay, those are two things I know about you.
Well, good evening, everyone.
Did I say Allison and not Christina? Allison Kraus is the singer. that
Yes,
I-
Allison,
that I said-
yes.
Christina.
Hello, can you hear me? Yeah. Well, thanks so much for being here. It's great to see, you know, people from the community interested in science. It feels good to be a scientist to see this audience. Before we get started, I actually want to ask you guys several questions. One is, what do you think is the largest organ in our body?
Skin.
Oh my gosh. Okay, so then the second question is, what do you think skiing does? What is its function?
Prevent you from leaking out.
Exactly, protection, right? Water doesn't come out. Pathogens, toxic compounds don't go in. It also has a lot of other functions, right? Touching, senses, temperature regulation, right? Skiing is where, well, vitamin D is made. Very good. So then the third question is, because it's so important, what do you think are the two most critical, important skincare routines or practices that we should have?
Washing. Sunscreen.
Very basic. I wonder if it's more for like social or for, but yes.
We appreciate that about you.
It is very important, fundamental. What else?
Sunscreen.
Sunscreen, right? Great. So you don't your skin doesn't prematurely age. You don't get skin cancer. What else?
Drink water.
Very good. Drink water. You get water to skin from within, lotions, so that all of that is to keep our skin moisturized so that we can have a good protective function, right? A good barrier. So you guys got 100%. So if you ever wondered, you know, this is how skin looks from the outside. So what does skin look from the inside? So if you cut the skin like this way and you kind of look this way, what you see is skin has multiple layers, right? The very top layer is epidermis. This? Yeah. So this is the epidermis. This is actually where the real protection is. And the epidermis is composed mostly of epidermal keratinocytes. This is a type of cells that is the major component of skin. And then you have the dermis, which is predominantly made of fibroblasts. You also have hypodermis, which isn't necessarily skin in a technical sense, but it's very important to attach the skin to the underlying bone and muscle and everything, okay? And as I said, this is really the business center of the skin. It's the epidermis, and it is a multi-layered structure. It contains this layer we call basal layer. That's where the stem cells are for you who are interested in stem cells. These stem cells can divide on a regular basis to supply the cells that gradually move up to the skin surface. As they move up, they change what proteins they make. We call this like a regulation of gene expression. They make different kind of proteins here versus here. And they change their shape, their behavior. Eventually, they become these sort of half-dead cells at the very top surface that are buried in lipids. This is the sort of brick and mortar structure on the top of our skin that seals the organism so that water doesn't leak out and pathogens don't go on. So it's a physical barrier, but at the same time, it's an immunological barrier because if pathogens do go in, then skin is able to mount immune responses because it has these resident immune cells. It can also bring immune cells from the body, other areas of the body, like the lymph node, so that it can mount immune cells, immune responses to kill the pathogen and remove cells that are damaged. Okay. And skin also have a lot of these appendages, for example, hair follicles, sebaceous gland, and sweat glands that each have their very critical functions. So when skin isn't functional, diseases occur. There are a whole wide array of skin diseases, but the two most common inflammatory skin diseases are psoriasis and atopic dermatitis. And you probably heard more of eczema, which is the other name for atopic dermatitis. So if you look at these conditions, they sort of look similar on the outside, but they actually have very different we call disease mechanisms, and they have very different disease manifestations. Psoriasis typically is sort of these lesions are kind of pink, red, they're scaly, and there is very fast cell turnover, so the skin can be very thick. And psoriasis can be linked to sort of even more serious conditions like psoriatic arthritis, and exactly what the connections are being studied. Atopic dermatitis, on the other hand, is more sort of related to conditions of allergy. And the skin or the lesions tend to be dry, cracked skin, and with a lot of itches. Okay. What are the treatments for these conditions? This slide, she's the expert, but I'm gonna just tell you about it. If you have questions, I'll direct you to her. But in the clinic, before we have the so-called targeted therapies, there are these various therapies that people use that include basically immunosuppressants. Steroids, they're immunosuppressants. They can be used topically or they can be used systemically. There are also use of drugs like methotrexate, cyclosporine, and to quench basically immune responses that are believed to drive these diseases. But there are issues with such old tools, and one is the benefit can sometimes be short term, right? And then there'll be remission, but then there will be relapses. The flares can come back. Steroids can, when they are used at high dosage or for a long time, can actually cause skin thinning and other side effects. If you use broad immunosuppressant, you can imagine that you can have whole body immune suppression, then making patients sort of more prone to being sick, right? Also, when patients are on these drugs, they may require frequent lab monitoring, and not all patients would respond. So we need better options. So what can scientists then do to help finding better treatments for these conditions? Actually, over the years, scientists have done a great deal to understand the pathogenesis of these diseases. By disease pathogenesis, what I really mean is what scientists are asking is what cells in the skin are driving these diseases? How do they start? How do they progress? If they undergo remission, what make them come back, right? So these are the kind of questions. Besides cells, what are the molecules, proteins, right? That actually are driving diseases. How do these cells talk to each other to orchestrate inflammation? These are the kind of questions, and there's a lot known now about the pathogenesis of psoriasis and atopic dermatitis. Psoriasis, people know that it's driven by abnormal immune responses. And in fact, it's driven by a specific type of T cells. These are immune cells. And there's this specific type called Th17 cells. And what these cells do is they can instigate additional inflammation. They can cause the epidermis to hypothicken and have very fast turnover. So knowing that, then what scientists can do is develop proteins. These are antibodies specifically that can block the function of a protein that is made by these Th17 cells, and that's called IL-17. So we have similar situation in atopic dermatitis. It's also driven by aberrant immune responses. And in that case, it's driven by another specific type of T cells called Th2 cells. And these Th2 cells make yet other sort of different proteins, specifically IL-4 and IL-13. So again, scientists developed antibodies that can block IL-4 or IL-13 function. And it's these antibodies that are now used in the clinic as biologics to treat psoriasis and to treat atopic dermatitis. And they've really revolutionized how patient care is for these particular diseases, okay?
Are those topical administration or do you take an injection?
Very good question. So they're not topic. Yeah, they're not topical. And that's the problem with them is when you have injections, basically it's systemic, right? They can cause side effects. Because these pathways could be functioning in other areas, other tissues. And they're also quite expensive. And also then patients, not all patients respond. So you nailed it, right? What we really need, I mean, we need these, but we also need a topical drugs that can actually be used in a more local manner that are effective, but not just steroids that does everything, but more evidence-based or mechanism-based therapies. And in fact, several years ago, a new drug, tapinarof, has received FDA approval, and it's actually the first and only FDA-approved steroid-free topical medication for psoriasis of all severity. And this drug is now also in clinical trial for atopic dermatitis. This is the structure of the drug. It's a small compound, and it's believed to function or to act through this protein called aryl hydrocarbon receptor. It's basically a protein that can bind a drug or a ligand, and it can go into the nucleus and regulate what proteins are produced, regulate gene expression, okay? And it's funny because this receptor is believed to actually be the target of active compounds in coal tar. And coal tar has been used for thousands of years, well, not for thousands of years, since thousands of years ago to treat skin inflammation. And later it was learned that active ingredient actually function through AHR. So sometimes nature does these experiments for us, right? Except we as scientists make them happen faster in the lab and with a more targeted kind of view. Okay, so and when you think about targeting topical treatment, one of the cells or cell types that we would want to target could potentially be the epidermal keratinocytes, right? Because they're on this outer skin surface and they're closest to the cream if you put anything on. But we don't really know a lot about how these keratinocytes are involved in inflammatory skin diseases. And that's a lot of what my lab does, is to try to use animal models to understand the role of epidermal keratinocytes in skin inflammation. And we've used models that are developed in the field to study these diseases. For example, we can use house dust mites and Staphylococcus enterotoxin B. We apply them to the skin of the mouse, and they can induce skin pathology that are similar to human atopic dermatitis. And this actually makes a lot of sense because house dust mites, they're in our houses, and their abundance have actually been found to be associated with sort of atopic dermatitis occurrence in some ways. And also people who have atopic dermatitis, they tend to have a lot of this bacteria called Staph aureus in their skin. And this toxin is made by Staph aureus. And you can imagine these agents sort of playing critical roles in disease pathogenesis. And we use that, take advantage of that knowledge to make mouse models and do our studies. And we've identified genes, specific genes that are active in the keratinocytes. And if you remove these genes from the mouse, we have technologies to do that. And when you do that, you get more disease, more severe disease. Similarly, we can use another drug called imicumod, which actually is used to treat like basal cell carcinoma in the clinic. But if you use that drug on the mouse skin, you induce psoriasis-like skin pathology. And again, we can use the model to identify genes or molecular pathways that are important in disease progression and try to target those. Okay, so.
Thank you so much, Xing. But with all of that being said, and Xing introducing us to psoriasis and eczema, there are still many skin diseases that are not well studied and really need better treatment. So as Xing kind of highlighted, in the last few years, in the last decade, psoriasis and atopic dermatitis have really come up with all of these targeted medications that have hit the market, and that gives patients more options. Now, when you think about dermatology, when you think about skin disease, what is one of those diagnoses that you always think about and you worry about? You've probably all heard about melanoma, right? Or skin cancer. And yes, today we're not talking about skin cancers, we're talking about rashes, right? But while skin cancer is very severe and needs appropriate treatment, rashes do as well. And we don't think about sometimes the burden that rashes have on a person's quality of life. And so sometimes in dermatology, we end up treating a lot more quality of life symptoms than we do actually curing people of a disease. So today we're going to talk a little bit about lichen sclerosus. And this will be a skin disease that I don't know if many of you have heard of. If you have, raise your hand. I'm curious. Okay, excellent. Okay. So we're going to talk a little bit about lichen sclerosus. We want to increase awareness of this and then talk about our collaboration together and our journey and why we think it's important to study this and potentially how this model could be applied to other skin diseases as well. So why does this matter? We're going to start with a patient story. So this was an article that was published in the British Journal of Dermatology, and it was a patient who very kindly volunteered to share their experience having lichen sclerosus, and then their clinician also kind of tag teamed and wrote about their experience. And so in medicine, we talk about the art of storytelling and how storytelling is very important and very cathartic. And I wanted to share with you just a little bit about what this patient shared about her disease. And so she basically said, hey, I had these unbearable symptoms of itching, burning, fissures, or cracks in the skin. And it became so painful that she could not wear underwear. And we're going to talk about where this disease tends to affect. And her online research pointed her to a diagnosis of this rare disease, lichen sclerosus. But because she had fear and denial, she delayed pursuing a diagnosis. And she had fears that she would develop vulvar cancer after looking this up online. She could no longer wear clothes comfortably, and she was crying from pain. All very, very severe symptoms, right? And she said, living with this is challenging. After 14 years, I can still not identify what triggers my flares. And we're going to talk about this a little bit later, but a lot of the patients who come in with these rarer skin diseases, they've gone almost a decade or longer undiagnosed and untreated. So think about being so uncomfortable and full of shame about your condition for 14 years, that really takes a psychological toll, right? And then she even said she was a nurse, and she said, despite being a nurse, navigating the healthcare system for treatment has been confusing and often dismissive. And I think this is something we see, and we were just talking about this with Leslie, I think this is something we see too often in women's health. Ultimately, so she and the clinician they wrote each of their pieces and they kind of teamed up at the end, and they said, I hope together we can better support those with understood, studied, and misunderstood conditions to enhance skin health for all. And that is really our goal in doing this research too. So let's talk a little bit about this underserved skin disease. So lichen sclerosus can affect any area on the body. And for all intents and purposes, it is an itchy rash. But in 85% of cases, it affects the genital skin, which is uncommon. Most of our skin conditions don't have a predilection for genital skin. They can occur anywhere. And for women, it most commonly occurs on the vulvar skin, and it's more common in women, and we're going to talk about that. You can see in this little kind of cartoon image we had published previously, there's these kind of white patches and plaques there. I don't know if you see that whitening, that porcelain white area. And you might say, that reminds me of a condition like vitiligo. I don't know if any of you have ever seen or are familiar with vitiligo. And it can look clinically like that. But the difference with this condition is there's a ton of inflammation or those inflammatory cells. And so what happens is that skin actually, for various reasons that we're trying to identify, gets thin over time. There's a lot of inflammation. So instead of it bending, it breaks, it tears, it cracks, and it leads to significant pain. And symptoms include that pain, that itch, soreness, and those tears in the skin. And you can understand why this is a really uncomfortable thing for people to come to their physician about, right? It's a super uncomfortable area. We don't talk about this area. Most patients come in and they worry, do they have a sexually transmitted infection? Is this something that's contagious? Is this something they could have gotten from their partner? And in introducing this disease to you, I want to tell all of you this is not. This has nothing to do with that. It has to do with various things we're trying to elucidate, but probably is an autoimmune disease, and definitely not contagious. And we know that this disease is associated with increased rates of depression and anxiety. It can affect intimacy, urination, and quality of life. And I think the biggest thing when we talk about vulvar skin disease, which I know I'm introducing most of you to today, it's not a common thing you hear about. But as we increase awareness, I hope that this becomes a more comfortable topic because it significantly affects quality of life, not just from a sexual standpoint. I think we minimize the disease if we talk about it from a sexual standpoint alone. A lot of these patients just want to be able to sit, want to be able to walk, want to be able to run, etc. And this is a different type of rash than psoriasis or eczema in that probably because of the area that's involved, if this goes untreated, it leads to scarring. And if we get scarring here, there are problems with urination, worse pain, etc. And there is about a 5% risk of developing a squamous cell carcinoma or a skin cancer if that inflammation goes uncontrolled, which if we think back to our patient's story, right? She looked up this condition, she thought that was what she had, and she became worried. Am I developing a skin cancer? So it's under-recognized. There's no FDA-approved therapies for this condition. And you might say, well, how common is this? You know, we talked about the prevalence of psoriasis and atopic dermatitis, and it's probably underreported, right? Because clinicians like myself are missing it. We're not diagnosing it. We're not asking patients about symptoms, and patients may not be disclosing to us they're having symptoms, but it's estimated to be around 0.7% of the population. In some gynecology clinics, that can go up to higher, like 3% of the population. It's, like I said, likely more common than we think and much more common in women. So you can see from this population pyramid, they highlighted in pink the women that were affected and in blue men by age. And what you see is that women are overall more affected. This is from just one study, but studies report that women are affected 3 to 10 times more than men, which is common with autoimmune disease, right? We see that more commonly in women than men. So why is it often missed? Well, you can guess now that we've talked about it. Symptoms are sensitive. There's a stigma around discussing vulvar pain, specifically vulvar itch. The other thing that I didn't mention is a lot of patients are worried that they're not using the right products or they're not cleaning the area or there's issues with that. So a lot of stigma around this. And then many clinicians are unfamiliar. So some studies show that patients may wait 5 to 15 years for a diagnosis. And I could go through there's a lot of challenges. Fortunately, some of our patients are very willing to help us with research, and we're so fortunate to work with them. And we asked our patient experts, like, if you could, tell doctors, if you could tell researchers what you need, what are some of the biggest goals for you, what would you tell them? And they said, we really need broad education. We need to increase awareness of this disease. And so you might say, okay, well, now we know what the disease is. There's no FDA-approved treatment, so what do you do? Like, what happens when a patient comes in? What are we doing moving forward? So high-potency topical steroids, which Xing kind of talked about, are the mainstay of therapy. And their topical steroids are often effective, but not always. Not every patient is going to respond to high-potency topical steroids, and topical steroids can have side effects, as Xing already mentioned. There's other therapies that we use all off-label, again, because there's nothing FDA approved for this condition. And the really important thing to know is that recurrence is common. It is a relapsing remitting disease. So we don't have a cure for it, which means patients will have to use some type of chronic therapy, basically lifelong. And so when you tell someone they have a disease, but then they have to use a topical steroid, maybe a couple times a week, basically forever, that can be really concerning, right? You hear about these risks with topical steroids. So what are other options for our patients? What do we do if our patients flare with topical steroids? And where do we go from here? So there's a lot of things we don't know about this disease. Why does it start? Which cells and molecules in the skin drive this condition? And if we had a better idea of that, could we develop better targeted therapies besides these broad topical immunosuppressants? Why does it keep coming back, especially in the same place, right? Like, why is it localized to that area? And then why does the scarring happen over time? Unfortunately, we have a lot of new exciting tools to study inflammatory skin disease.
So, with the questions that Christina brought up, like how do we really address these? The one thing that is super exciting in the last 10 years or maybe less is we now are able to actually look at a tissue and we can break the tissue into individual cells and we can look at what genes are active in each individual cells and we can look at thousands of them at once. And this technology is called single cell RNA sequencing. Basically, what we can do is we take the tissue, we break it into single cell suspension, pass it through this special platform where the cells are sort of given their individual like address or postcode. And then we can sequence the cells, but because they have that address in them, then we can look at their sequences in a collective manner using computational methods and sort of high dimensional type of analysis. And we can group these cells into their relative kind of relevant cell types and then look at their gene expression programs. So this allows us to look at what is different between disease and normal skin, right? So another technology that is sort of on the, not like it's really getting a lot of scientists excited is called spatial transcriptomics. And here, what's the difference from single cell RNA sequencing is for single cell RNA sequencing, we sort of took all the single cells in a tube and then we try to analyze them, but we don't know where every cell is in their original location. But with spatial transcriptomics, we get to look at the activity of each gene in its native tissue environment, right? So if we have signaling genes and we know they're active, we have a ligand, we have a receptor, then we would be able to see, hey, are these cells close together so that they have the likelihood of talking to each other via that kind of signaling mechanisms, right? So we have different varieties of how to do spatial transcriptomics. But the bottom line is we could analyze like 5 genes at once, or we could analyze 5,000 genes at once, or we could just have sort of this map and spatial map of barcodes, and we can put the tissue on top and then sequence the whole genome of the cells on their in their native tissue microenvironment. So with those kind of techniques, we apply them to analyzing patient samples from lichen sclerosis and patients. And as Christina said, we were very fortunate to get volunteers who actually donated their skin so that our research is possible. We compared patients their lesional skin. This is where there's full-blown disease. Compared with their non-lesional regions. These are surrounding regions where disease is not yet apparent. And our hope was to find differences that can, differences in cell types, differences in genes that are active, and see the ones that are in the lesional skin might likely be candidates that would drive disease in some way, right? That's kind of our logic. And with spatial transcriptomics, we're able to generate images like this. This is an example. There's a lot more there's a lot more cells we can see, but this is an example of that. Here you can basically see 3 different cell types, you know, the epidermal keratinocytes. This is the stem cell containing epidermal keratinocyte compartment. You could see melanocytes in red here that are part of the epidermal structure. You can see endothelial cells, right? Some deep in the dermis and some actually go into these special structures in the skin. You could also look at individual signaling genes. The dots here represent a specific inflammatory gene that is active in only specific compartments. For example, it's now here only in the epidermis or around the endothelial structures. And again, with this type of information, we can compare what is different between a lesional skin versus a non-lesional skin, or what is the difference between lesional, but non-lesional skin from a patient versus a skin from a healthy adult, a healthy subject. Okay. And with that type of technological advancements, we were able to make several key findings related to the mechanism of lichen sclerosis. And at a higher level, one thing we're finding is this is not just like one type of cells that are involved. Multiple cells, type of cells are involved. Keratinocytes are affected, fibroblasts affected, melanocytes are affected. Melanocytes are actually pigment producing cells in our skin. So related to the similarity of vitiligo, this is interesting to find melanocytes having specific changes that might contribute to disease pathogenesis. Of course, we also see immune cells. So that's the one thing. This is a multifaceted, multi-component involvement. And also these cells actually talk to each other in different ways in the lesional skin versus the non-lesional skin. And the other thing we're finding is when we look at different patients, we actually see differences, even though there are common features at the cellular and molecular levels. Patients, these samples also have differences in the specific changes that they display. So we call this heterogeneity, patient to patient heterogeneity. Is it because the disease in different stages? Is it because there are fundamental differences or subtypes that the disease comes about? And those are the interesting questions that we're hoping to address. And in terms of specific findings, we're finding, for example, keratinocytes. This is the cell type of my love. They are stressed and inflamed, and they're potentially sending signals that could activate the immune cells. We're finding fibroblasts, actually, they're not producing enough collagen comparing to their more healthy counterparts. And this is surprising to us because when you think about lichen sclerosis, based on the very limited evidence in the field, you think about scarring, right? You think about fibrosis. That's when you would think more about like an overproduction of collagen. But using this technology, what we're seeing is actually underproduction of collagen. So that should change our thinking about how do we really approach this disease when we think about treatment. Not surprisingly, we see immune cells being active. We actually see these immune cells that like to go around and kill surrounding cells. But what's interesting is these immune cells appear to be stuck in a chronically active state, and they even become sort of exhausted. So does that inform us in any way in terms of future treatment? That's something interesting to think about. And the other finding we thought was important is even when you look at the non-lesional skin, you're already seeing signs of aberrancies. Even though the lesions or the disease is not full-blown, there's already changes that are suggestive of disease onset or disease initiation. So this would be important when we think about potentially preventing disease from going bad, right? And finally, I think one of the most important findings is we see multiple patient samples displaying a strong signature suggesting they have activation of this pathway called JAK-STAT. JAK-STAT is actually an infamous signaling pathway that's really made it to kind of people's mind in terms of skin disease, because it's involved in a number of autoimmune type of skin diseases. And what do these findings actually mean for the patients? Christina is going to tell you about.
Thanks, Shane. Okay, and so why are these findings important? Why do we focus on this JAK-STAT pathway? Well, as Xing had already alluded to, there are, with advances in science of skin, we found very many different molecules that can be altered in inflammatory skin disease. And if we have something that is either a directed therapy like a biologic or a small molecule inhibitor, we can provide potentially more targeted therapy to patients to improve control of their skin disease, and then decrease side effects. And the great thing about the JAK-STAT pathway being involved in this disease is that there are a number of JAK inhibitors that are available, that are on the market for various autoimmune skin conditions, and autoimmune conditions in general. So rheumatoid arthritis, they're being studied for lupus, they're being studied for all kinds of conditions, and we already have them approved for eczema, which is fantastic. So if we see that, are there medications that currently exist that we can repurpose for this disease, or can we find new JAK-STAT inhibitors that might be more efficacious? That could disrupt the inflammatory cycle and may reduce itch and pain. And so what it means for patients, I think, today what we wanted to talk about was an underserved disease, really validate the symptoms that occur with this and increase awareness. Really, there is a need for early diagnosis, and this is something we need to go out and educate our medical students about and clinicians about, and then get all of our scientists to help us identify more targeted therapies. What we don't have, and Xing talked about the animal models that exist for psoriasis and eczema and other types of disease, we really don't have an animal model for lichen sclerosis. So really probing what's going on mechanistically is quite a bit more challenging. And we do need that and future clinical trials to really determine what is best for our patients. So these are our acknowledgements, and we're very fortunate to have UCI supported so much of our work throughout this time. And thank you for your attention.
Fantastic. I love because you can see all the years of collaboration with this talk and the integrated talk. That was so great.
Yeah,
Thank you.
we work together quite a bit. It's a lot of fun. And I should say that this actually was an area we didn't really have official outside funding, and it's the university, various units that realized how important this is and provided seed funding so that all of this is possible. And Leslie is directly responsible for a huge chunk of the funding that has gone into this study. So it's
Wonderful results. And it was just, you should say, it was just published this year in Journal of Dermatology.
yeah, it was published, and also the journal arranged a news and views kind of commentary on it, because it's an ignored disease, and the journal also recognized the importance of increasing awareness. So we're very, very happy.
Super cool. That's wonderful.
Any questions?
Go ahead.
I was curious to see, did you see upregulation of JAK-STAT in the non-lesion cells as well? And I guess my second question is, the JAK-STAT pathway is expressed in pretty much every cell. So how do you know that you're going to have targeted inhibition just for the disease cells and not off target?
Are you from the community, or are you in grad school?
No, I'm in developmental cell biology. Yeah.
Okay. Okay, no wonder. Great.
Have a mix of audience.
Okay. Great questions. I mean, we could look at where JAK-STAT is active, right? And the comparison that led to the conclusion of increased JAK-STAT is comparison between lesional and non-lesional skin. So we know it's higher in the lesional skin comparing to the non-lesional skin. And we also have ways of actually measuring kind of the downstream, phosphorylation of proteins, and we know that it's more active in the lesional skin. but it's a good question when you target and you use JAK-STAT inhibitors in the patients, how do you know which cell to target? We don't know. The use for rheumatoid arthritis, lupus, also you don't know. But the idea is if we look at actually the skin cells, we can have computationally, we have methods to predict exactly where the activity is the highest. And our hope basically is once the drug goes in, it targets those cells, whereas others are not affected in any way. But great questions. Okay. I would love to have you work in my lab.
I noticed on one of the graphs earlier on, like when it was the female versus male, that there was like a spike in like early 20s and then later on. I was wondering, have you looked into like if there's any correlation between like puberty and then like post-menopause?
Yeah, that's a good question. So it used to be thought to be a disease of estrogen deficiency. So it was thought that it would be there pre-puberty, so kids would get it. And then once puberty started, it went away. And then menopause, when it's low estrogen again, it comes back. By far and above, most patients who have this, most patients who have vulvar lichen sclerosis are postmenopausal. But what we've seen in recent years, and we don't know if there's actually an increased incidence or we're just better at diagnosing it earlier, but we're seeing it much more commonly in patients of reproductive age. So if you look back at older studies, and this is actually a slightly older study, you will see that they really say there's kind of those two peaks before puberty and then in menopause, but now we know it can affect anyone of any age. It's thought, there's some thought that maybe dropping estrogen is another trigger to make it worse or make it more symptomatic, but does it truly like promote disease onset? That's probably unlikely. It's probably just one of the factors in making disease more recognizable, symptomatically and clinically. It's a great question. Yes.
With the same subject matter, looking at the peaks for male versus female around 20, and then continuous drop for males, but not the same pattern for the female.
Yes.
And the question is, why do we get nice drop as you age? And then in case of males, it just keeps on dropping, whereas in case of females, you now have rising as you have increased in the age.
Yeah, I think in males, it is overall staying a little bit more, it's more similar right throughout, whereas in females, you definitely see it rising as you age. But the other thing is, I don't know if this is true because there's not enough studies, but from a clinical standpoint, it gets easier to diagnose the farther along it progresses, which is a terrible thing, right? But so, sometimes by the time I've seen a patient, I'm like, we're 10 years, like we should have diagnosed this 10 years ago, right? Often, so one of the thoughts behind it being less common in males, right? One, it's an autoimmune disease, so probably less common in males, but two, in patients who are circumcised, lichen sclerosis is less common, and one of the treatments for lichen sclerosis in males is circumcision. So sometimes there's actually cure in male patients with circumcision.
But separate question
Yes.
from that is, some of the skin diseases are treated with various wavelengths of light. For example, lasers. Are those applicable in this situation? And the other one is related to it with electric fields. Low intensity electric fields, whether they do impact the electrochemical processes.
Those are great questions, and I feel like your first question in regards to light-based therapies, could be a lecture on its own. So in the past, we used specific wavelengths of light, like narrowband UVB therapy. The risk with it on the genital area is that the genital area is an area we actually protect when we have patients come in for narrowband UVB for psoriasis and things, because we think there's an increased risk of skin cancer. So we're a little bit wary about using that as a treatment. Now, in recent years, These CO2 or erbium YAG lasers, which are used for resurfacing and scars and are fantastic lasers, have started to be used for lichen sclerosis. And there are some studies, and there is data that suggests that they do help with lichen sclerosis, but they are not a cure. The problem is they're not covered by insurance, and they're usually thousands of dollars. And so sometimes they're marketed to what I would say is a vulnerable population, because these are women seeking treatment who have been untreated for a long time, have severe symptoms, and they can be marketed as a cure, and they're not a cure. Patients still require maintenance therapy. So yes, my long answer, yes, they probably do have a role, but they're not a standard of care by any means, and not necessarily recommended. It depends on the patient and the degree of inflammation. And then to your second part, I do not know the answer to that. I stopped at the laser.
I was just curious, just thinking about models, like, could you envision using the keratinocytes or the fibroblasts or anything as some kind of cell-based model, or reprogramming stem cells, or?
Yeah, so we've thought about this a lot. We kind of wanted to propose organoid models using keratinocytes and fibroblasts, because we think those are two drivers of disease. But then how do we incorporate the immune cells into our model? I'll turn it over to Xing, who has much more experience with this.
Yeah, I mean, that's a really a very important question. Is to build it, I think our problem right now is we just cannot, there's not a good way to put immune cells into the keratinocytes fibroblast model, because they require very different nutrients and growth factors for growth. So putting all of them all together and assemble a skin that has all the right component is very
Wonder
challenging.
if you could use like conditioned media from immune cells or something, anyway.
That would be a possibility. That would be, the other possibility is actually to take skin explant,
Yeah.
right?
Yeah,
So you can like take from vulva skin and have, the problem with those is you can't really culture them for very long, maybe a couple of weeks at the most. So that's kind of an issue. You can try to do mouse models, that would be a possibility, but the problem sometimes is when, I know like for this other disease called HS, or how do you say it?
Hidradenitis suppurativa.
Yeah, it's a devastating disease and it creates that, yeah, very deep like tunnels in the skin. When people try to study it using animal models, they take the patient's skin, put them onto immune compromised mouse, then the disease is no longer, it does something else, it doesn't replicate the disease. So these are the kind of challenges that we would just have to deal with. I think stem cells has a lot of potential,
Yeah.
right? So if one does like IPS
right.
and differentiate them into different kind of lineages and put them together, maybe,
Yeah.
right, there
That's
would be
possible.
ways. So that would, yeah. And I also believe, because this disease, I think there's a strong atrophy component there that people, I mean, we are talking about inflammation, but I definitely think there's atrophy. And there is depletion of epidermal stem cells. Clearly, that's what
Yeah.
we're seeing. So if there are good ways of somehow
Getting
putting
stem cell.
stem cells into the patient's, right, injection or something, I think it's gonna recreate a healthier microenvironment that just is corrective. But,
That's very cool. Yeah.
So you mentioned this is already a rare disease. It is hard to diagnose, and often diagnosed late because patients are hesitant to come in. I'm just wondering, like, how you think that might impact, like, the ability to perform clinical trials on these eventually drugs.
Yeah, it's a great point. So we did a clinical trial here, and we actually have one opening in the near future. And I have to say, this is like the most motivated and engaged patient population ever. Like, they so badly want to help with research in this disease, because I think there's so much suffering associated with it. So I don't know, at UCI, we're really fortunate because we have the best patients. And so there was no trouble recruiting for that first clinical trial. And in my experience talking to colleagues at other institutions, they have similar experiences. So luckily our patients really want to do research and are really motivated to do it.
Is that with the JAK inhibitors? Or you doing-
Yeah, with the topical JAK.
Cool. Okay. Oh, one more question.
Sorry,
and then-
have you guys looked into at all like collagen supplements for patients? Just because I know as you get older, like you you lose your collagen like little by little. And obviously like the patients, as they get older, there's like a bigger spike. So is that something that's like important or like, have you looked?
Yeah, it's a great point. There's a lot of stuff on, in the media about collagen supplements and their use for various skin diseases. As far as I know, there's not been any studies on collagen supplements for lichen sclerosis specifically. The tricky thing about collagen supplements is when you take them orally, they're digested in the gut. And so you're like, how is that collagen like truly getting into your skin? So then that comes into like some people are using fillers and they're taking like fat from other areas and injecting it there, and there's very little evidence for that, but it is a good point. If there was some way we could help, maybe not increase collagen, but make healthy collagen for people, that would be great. It's just as far as, with collagen supplements, the way they are, that is not the avenue right now, but hopefully going forward, we'll have a better means.
Well, thank you so much, but this was wonderful. Thanks.